Welcome students. Today we are going to throw light on structure of stamen and male gametophyte. The stamen is the male reproductive or pollen producing organ of the flower. A typical stamen consists of a lower sterile part called the filament and an upper fertile part, the anther. Anthers are most commonly two-lobed and are attached to the filament either at the base or in the middle portion. The sterile tissue between the lobes is called the connective. The anther shows great variety of forms, but typically it is two-lobed and the lobes are called anther lobes. Each anther lobe has a pair of pollen sacs or microsporangia. Thus, characteristically, each anther has 4 microsporangia. More than 4 microsporangia in an anther is rare, but less than 4 is frequent and characteristic of many taxa. The microsporangia form sacs or pockets or locules in the anther. The two separate locules on each side of an anther may fuse into a single locule. The young anther is a homogeneous mass of meristematic cells surrounded by an epidermis. Soon groups of hypodermal cells in each of the 4 corners become distinguished from the surrounding cells by their large size, dense cytoplasm and prominent nuclei. The cells, which are arranged in plate-like or crescent-shaped vertical rows, form the archosporium of the anther. The number of archosporial cells varies in different taxa. In families Malvaceae and Asteraceae, there is only a single vertical row of archosporial cells, whereas in some members of Lamiaceae, such as Mentha, there is a layer of archosporial cells at each corner. The archosporial cells enlarge radially and divide periclinally, resulting in outer primary parietal cells and inner primary sporogenous cells. The primary parietal cells undergo repeated periclinal and anticlinal divisions, giving rise to 3 to 5 concentric layers, which eventually form the anther wall. The primary sporogenous cells may directly function as microspore mother cells or undergo several mitotic divisions and then function as microspore mother cells. Now, we will discuss mature anther wall and sporogenous cells separately. Mature anther wall. The mature anther wall comprises an epidermis followed on the inside by a layer of endothecium, 2 or 3 middle layers and a single layered tapetum. Now, we will discuss them one by one. Number 1, epidermis. This is the outermost wall layer of the anther made up of tangentially stretched and flattened cells. In xerophytic plants, the epidermal cells are stretched to such an extent that they lose contact among themselves and appear as withering remains in a mature anther. Stomata may occur in the epidermis, as in elengium. The epidermis performs its usual protective function. In Zeoxene longilabris, the epidermal cells stimulate tapetum. They may even become binucleate. An unusual feature is the development of fibrous thickenings in the epidermal cells of Arceothobium, forming exothecium, which is characteristic of the microsporangia of gymnosperms. In Morus, the epidermis degenerates at maturity. Number 2, endothecium. This is the second or subepidermal layer of the anther wall. Mostly there is a single layer of endothecium, but sometimes, as in Coccinea indica, it is two-layered. The endothecium originates from the parietal layer and is present in the protuberant part of the anther. A similar tissue, when present on the inner side, is formed by the cells of the connective. In triticale, however, the entire ring of endothecium is formed by the parietal layer. The cells of the endothecium are radially elongated and attain their maximum development when pollen grains mature. The radial and inner tangential walls of these cells have characteristic fibrous thickening bands. This thickening is made up of alpha cellulose with a little pectin and lignin in some cases. Usually, these bands are U-shaped or ring-shaped, but in Melothria medarspatana, they occur in the form of two overlapping spirals, and in Momodica carentia, transverse bands of thickenings are present. These bands are formed at the expense of the starch stored in endothelial cells. In those anthers which dehisce longitudinally, the endothelial cells at the junction of two pollen sacs of an anther lobe lack thickenings. In some Clastogamous species where flowers do not open, and in the members of the Hydrochaerataceae, fibrous bands are altogether absent in the endothecium. The endothecium of those anthers which dehisce by apical pores also lack thickenings. Endothelial thickenings are also absent in species of Musa, Sesamum, Annona, Ipomia, etc. But in these taxa, the inner epidermis has deposition of cutin and lignin. The presence of fibrous bands, differential expansion of inner and outer tangential walls, and hygroscopic nature of endothelial cells play an important role in the dehiscence of anthers. Number 3. Middle layers. It is the third wall layer of the anther. The number of middle layers generally ranges from 1 to 3 and are lying next to endothecium. The cells of middle layers are ephemeral and degenerate almost completely before the pollen mother cells undergo meiosis. Sometimes these layers degenerate soon after meiosis, as in Melothria medarspatana, Momodica carentia, etc. Occasionally, one or more middle layers persist for indefinite period of time, as in Coccinea indica and species of Ranunculus, Holoptilia, and Lilium. The middle layers are absent in the members of Lemniaceae and Najadiaceae. The outermost middle layer in Gloriosa is endothecium-like fibrous bands. The middle layers may store food material in some taxa which is utilized by pollen mother cells. Number 4. Tapetum. It is the innermost wall layer of the anther. Usually it occurs as a single layer around the sporogenous tissue and is characterized by the presence of dense cytoplasm and prominent nuclei. Sometimes the cytoplasm of tapetal cells is pigmented. For instance, red-brown pigments are present in apple and violet pigments in anemone. The tapetum attains its maximum development at the tetrad stage of microsporogenesis. The tapetum is of considerable physiological importance because all the food material to the sporogenous tissue passes through it. Tapetum is present in the form of homogeneous layer which completely surrounds the homogeneous tissue. In some taxa, however, a dimorphic tapetum is present. For example, in Nigella damascena, the tapetum on the protuberant side of the anther formed from the derivatives of the primary parietal cells is called P tapetum, and on the connective side, where it differentiates from the cells of connective and tapetum between the two pollen sacs, is known as C tapetum. The cells of C tapetum are relatively large than cells of P tapetum. In triticale, however, the tapetum originates solely from the parietal layer and is homogeneous. Sometimes the tapetum may also arise from the sporogenous tissue, as in Antirrhinum majus. As the sporogenous cells undergo meiosis, the nuclei of tapetal cells also divide, and as such, the cells of mature tapetum are usually multinucleate. In some orchids and cucurbits, the cells of the tapetum remain uninucleate. Now, we will discuss sporogenous tissue. The primary sporogenous cells divide and redivide mitotically to form a mass of sporogenous tissue. The last mitosis results in the formation of microspore mother cell or pollen mother cell, which undergoes meiosis. Thus, the meiotically dividing sporogenous cell is the pollen mother cell. Each microspore mother cell undergoes meiosis and forms 4 haploid microspores. The process of formation of microspores from sporogenous tissue is known as microsporogenesis. All sporogenous cells of an anther are capable of forming microspores, but usually some of these cells degenerate and provide nourishment to the active sporogenous cells. In some Gentianaceae, the tapetum is not well developed, and then the nutritive function is taken up by sporogenous tissue. Thus, during microsporogenesis, the nucleus of each microspore mother cell undergoes meiosis or reduction division and ultimately gives rise to 4 haploid nuclei. These 4 nuclei are arranged tetrahedrally and are soon invested with cell walls. These are now called microspores or pollens. These soon dry up and become powdery while the tapetum becomes absorbed. The anther now becomes a dry structure. The partition walls between the sporangia are destroyed and the microspores are soon liberated by the dehiscence of the anther. During meiotic division of microspore mother cell, the wall formation may be of successive or simultaneous type. In the successive type of wall formation, a cell plate is laid down between 2 daughter nuclei after meiosis first. This wall gradually extends towards the periphery on both sides, dividing the cell into 2 equal halves. Each cell of the dyad thus formed undergoes meiosis second. The wall formation between the 2 daughter nuclei occurs in the same way as after meiosis 1. This results in the formation of a microspore tetrad. The second meiotic division may not be synchronous in both the cells of the dyad. The successive type of wall formation is found mostly in the monocots. In the simultaneous type, there is no wall formation between the 2 daughter nuclei after meiosis first, and as such, instead of a dyad, simply a binucleate cell is formed after meiosis first. The 2 daughter nuclei then undergo second meiotic division, which is usually synchronous. At this stage, 4 daughter nuclei lie in the common cytoplasm. The wall formation begins in the form of 4 peripheral constructions which gradually extend from periphery towards the center. These constructions eventually meet at the center of the cell and divide the cytoplasm into 4 haploid microspores. The microspores of a tetrad are separated from each other by callose wall. There is also no connection between the microspores of different tetrads in the anther locule. However, in some orchids, microspores do show cytoplasmic connections. The 4 microspores formed from a microspore mother cell are usually arranged in tetrahedral or isobilateral tetrads, which are formed as a result of simultaneous division. The 4 microspores lie at the 4 corners of a tetrahedron. When seen from an angle, only 3 microspores are visible and the 4th lies at the back. In isobilateral tetrads, which are formed by successive divisions, the 4 microspores are arranged in one plane. Besides tetrahedral and isobilateral types, sometimes microspores may be arranged in decussate. For example, species of Magnolia, Crocus, and Atriplex. T-shaped, for example, species of Aristolochia, Butomopsis, or linear, for example, species of Halophila tetrads. The microspores of a tetrad usually separate from each other as the anther matures. But in species of Drymus, Annona, Acacia, Drosera, Typha, Elodia, etc., they don't separate even at maturity, and even the tetrads are stuck together in groups which may contain as many as 64 pollen grains. Such groups are called compound pollen grains. In Asclepiadaceae, all pollens in a pollen sac are united in a single compact mass known as polonium. Polonia are also formed in Orchidaceae, but in certain members of this family, the polonium is less compact as it comprises smaller groups of pollen grains. Such loose groups are termed maculae. The pollen grains of a maculum are loosely joined together among themselves by means of a Vicined thread in Ericaceae and Amphitraceae. The pollen grains are permanently united in tetrahedrons. Now, we will throw light on male gametophyte. First, structure of pollen grain. Pollen grains are the male reproductive structures that develop from the diploid mother cells in pollen sacs of the anthers. They are also known as microspores. The microspore is the mother cell of the male gametophyte. Typically, each pollen grain is a haploid and unicellular body with a single nucleus. It is usually round or oval or polyhedral in shape with 0.025 mm to 0.125 mm in diameter. There is a tiny mass of protoplasm surrounded by a thick cell or spore wall which is differentiated into 2 layers, the outer exine and the inner intine. The exine appears first as a thin layer but later becomes considerably thick. The exine is chiefly composed of sporopollenin, a substance considered to be the oxidative polymer of carotenoids and carotenoid esters. It is a tough substance providing resistance to physical and biological decomposition and thus checks natural decay of pollen grains. Preservation of pollen grains during fossilization is due to the presence of sporopollenin. The intine is composed of pectin and cellulose. The fibrils of cellulose are arranged parallel to the pollen surface. The intine is usually thicker under the pores, and at these points it also contains enzymatic proteins. In insect pollinated species, the surface of the pollen grains is covered by an oily layer called pollen kit. The sticky nature color, and smell of pollen grains are due to its presence. The substance necessary for the synthesis of pollen kit are secreted by the tapetal cells. Although definite function of pollen kit is not known, it is believed that it helps in attracting insects, protects pollen from ultraviolet radiations, and its sticky nature helps in sticking pollen to insect body. The cytoplasm of pollen grain contains dictyosomes, mitochondria, and endoplasmic reticulum. Pollen grains are densely cytoplasmic as long as they are in tetrads, but soon after their release from the tetrad, they undergo considerable enlargement and as a result of their cytoplasm becomes highly vacuolated. Now, development of male gametophyte. The microspore or pollen grain is the mother cell of the male gametophyte. While still in the anther lobe, pollen grains begin to germinate. Before mitosis, the displacement of the nucleus from center to one side occurs. This is followed by vacuolation. The first division of pollen grain results in two unequal cells, a larger vegetative cell and a smaller generative cell. Initially, the generative cell is attached to the intine of the pollen wall, but later it gets detached from the pollen wall and lies free in the cytoplasm of the vegetative cell. Eventually, the vegetative cell develops into pollen tube and the generative cells form male gametes. Of the two daughter nuclei formed in pollen, the one lying towards the pollen wall is the generative nucleus and the other functions as the vegetative nucleus. A thin, delicate, curved wall is formed between the two nuclei, which cuts off the generative nucleus in a small lenticular cell against the wall of the grain. It becomes free from the pollen wall after a short time, and then this biconvex cell gets surrounded by the cytoplasm of the vegetative cell. The shape of the generative cell may become ellipsoidal, vermiform, fusiform, or lenticular at different stages of development. The elongated form of generative cell, however, facilitates its passage through pollen tube. The nucleus of the vegetative cell is spherical or irregular in outline with thin chromatin material. It has one or two nuclei. The cytoplasm of the vegetative cell contains ribosomes, rough endoplasmic reticulum, plastids, active dictyosomes, mitochondria, fat, and starch grains. RNA and proteins are also present in sufficient amounts. The vegetative cell normally does not divide, although it is capable of DNA synthesis. The wall of the generative cell is regular in outline but looks quite irregular on closer examination. Its texture is relatively uniform and may show occasional small electron opaque structures. There is no cytoplasmic connection between the generative cell and the vegetative cell. The relatively dense cytoplasm of generative cell contains mitochondria with conspicuous internal membranes, endoplasmic reticulum, dictyosomes, and abundant free ribosomes. In species like Oenthera hookeri, the generative cell also contains plastids, but they do not contain starch. The nucleus of the generative cell occupies most of the cell volume. The nuclear envelope has numerous pores in its double membrane. The male gametes are formed by mitotic division of the generative cell. This division usually occurs before the pollen is shed from the anther, but in some species the generative cell divides only after the pollens have been shed. In most of the plants, the division of the generative cell in the pollen tube is characterized by the absence of metaphase plate and spindle formation. Many studies have shown that even if pollen are shed at two-celled condition, the generative nucleus is already in prophase stage and the process of division merely continues in the pollen tube. Thus, depending upon the division of the generative cell, pollens may have 2 or 3 cells at the time of their release from the anther. Two-celled pollens remain viable for a longer period and can be easily grown in culture medium. On the contrary, three-celled pollens remain viable only for a short time and they cannot be grown in culture medium. Mitotic division of the generative cell results in the formation of two non-motile male gametes. Each male gamete consists of a large nucleus surrounded by a thin sheath of cytoplasm, externally limited by a cell membrane. In most of the angiosperms, the cell membrane is not covered by cell wall, hence the male gametes are naked. The nucleus of the male gametes is bound by a conventional unit membrane. The cytoplasm of the male cells contains mitochondria, dictyosomes, ribosomes, and microtubules. The microtubules probably help in maintaining or altering the shape of male gametes as they course down the pollen tube. Functional plastids are generally absent in the male cells. The two male cells of a gametophyte do have physical contact with each other. Such a contact is important as it helps in coordinated development of both the cells. It is also important from the point of view of double fertilization. Dear students, this is all about structure of stamen and male gametophyte. Thank you very much.